Spatial color coding in V1, Part 2. We have shown that coupling spatial coding with color coding in V1 gives us large double opponent cells in the low spatial frequency band, and these cells are often insensitive to orientation because they occupy this isotropic frequency band region. And we also have orientation tuned cells with resistive field very small that are insensitive color in high spatial frequency band. And we also have cells simultaneously tuned to color and orientation in the intermediate frequency band. So their resistive field sizes are also intermediate between these two extremes. And we say that these cells are tuned conjunctively to color and orientation. And we note that for this to happen, these chromatic contrast sensitivity curve need to be wide enough to cover a large enough frequency band to include not just the lowest frequency band where neurons are untuned to orientation, but slightly higher frequency band. However, if we are in a dimmer environment, the contrast sensitivity curves will adapt like this. Due to a smaller signal to noise in this environment, frequency cutoff, both in the chromatic channel and the luminance channel will occur at smaller frequencies compared to previous case in a brighter environment. And this in particular forces the chromatic contrast sensitivity curve to be only substantially non-zero in the lowest frequency bands. And this means that although we might still have these double opponent color cells that are insensitive to orientation and orientation tuned cells insensitive to color, we will have fewer or no cells tuned to both orientation and color. And this may be why these type of V1 neurons have been found frequently in some experiments, in some literature papers, but less frequently in other experiments. It is likely that in some experiment the environment is brighter, the visual display is brighter with different equipment to display visual stimulus inputs than in other labs. V1 neurons tuned simultaneously to color and orientation are likely the basis behind the color effect, a color adaptation effect that is contingent on the orientation of the inputs. For example, if we look at this green horizontal stripes and this red vertical stripes by looking here and back and forth, presumably the neurons tuned to green horizontal are being adapted, and also the neurons tuned to red vertical are also being adapted during this process, so that after sufficient adaptation, perhaps 1 minute, these neurons become less sensitive or less responsive. And they become less responsive because, as we have learned from efficient coding, the gains are smaller for channels with higher signal powers. So being adapted to red vertical and green horizontal make these channels have higher signal power, therefore the gain become less, and as the basis for adaptation. So then after 1 minute or so, if you look at this colorless stripes, one is likely to perceive in this horizontal stripy region as pinkish and this vertical stripy region as greenish. This is because in this horizontal region, the green horizontal neurons are less responsive compared to red horizontal neurons, and so therefore looking more pinkish. On the other hand, here the red vertical neurons become less responsive. They are less responsive as the green vertical neurons, so therefore this looks more greenish. We have seen by using a simple case with only 2 cone types that there can be a diversity of neural types in terms of the relative spatial mixture of red versus green sensitive areas within a single resistive field. When there are three cone types, red, green, and blue, there will be a diversity of preferred colors among the V1 neurons. This is because there are three cardinal directions: luminance, red-green chromatic contrast, and blue-yellow chromatic contrast. So there are two orthogonal chromatic contrast directions. And there are three filters, one for the luminance channel, one for the red-green channel, and one for blue-yellow channel. And these three filters for any scale, okay, low frequency or high frequency scale, are then multiplexed in three-dimensional cone space to arrive at three populations of neural responses, O1, O2, and O3. If the original cone decorrelation matrix is K0, and here the multiplexing matrix is the inverse of that, and let these three filters be described by these three Gabor functions with the gains according to these contrast sensitivity gain curves and according to whatever the frequency band, the mean frequency there is for this gain. Now let's note that these two gains, one for the red-green chromatic channel, the other is for blue-yellow chromatic channels, they are not necessarily the same. They're not the same, okay? Even though to avoid clutter, we just use one curve to just to visualize them. They are actually different, even though they're both relatively lower pass compared to this band pass filter. Then, for the first two output neurons in this multiplexed results, we will have this function describing the red, green, and blue reciprocal field for each of these neurons. Okay, so let's say for one neuron, this is the amplitude, and that is a phase. A is the amplitude, phi is the phase for the red cone, amplitude and phase for the reciprocal field Gabor filter for the red cone, and this is for the green cone, and this is for the blue cone. Now remember, this alpha is a parameter that we use in this is actually a value used in this matrix. We have learned that before. But this is a very complicated mixing of these three filters according to the gain and phase. Gain and phase for the luminous channel, gain and phase for the red-green chromatic channel, gain and phase for the blue-yellow chromatic channel. So this can be a very lots of diverse possibilities depending on these phase values and gain parameters. For example, let's observe a very simple case when this gain for the luminance channel is negligible, 0, let's say, okay? It could be in a low spatial frequency where this gain is much weaker than the gain for the chromatic channel. So these two filters are more significant than this filter. If so, then we can just take these gains 0 for the luminance gains or 0. Okay, and that means we can focus on these parts contribution to the red, green, blue inputs of these cells. Now let's take another simplification that the phases, okay, the phases for the red, green, and blue, yellow channel Gabor filters are the same, and let's say they are equal to 0. And that means these exponentials are equal to 1. We can also ignore them. And therefore, we only look at these factors in the pink box, and these factors in the magenta box, and this factor in the blue box. And now, let's see. Okay, these have two signs because we're looking at O1 and O2. So let's look at the O1 output. So therefore, we take this top sign. So it's positive sign here and negative sign there. So positive, 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 these three positive, same sign and combined together. Here's a negative, negative, and these the opposite sign combined together. Okay, and so therefore we see that these three positive terms, they are a big factor for the red cone input and some little for the green cone input. So this is an orangish input sensitivity, and here is a bluish and a greenish put together, it's a cyanish sensitivity, but in the opposite sign to that. So therefore, this neuron should exhibit an opponency between orange color and cyan color. So therefore, some parts of the receptor field may be excited by orange and the other parts of the receptor field excited by cyan, and vice versa. Note that we are being creative here in naming color as orange and cyan. You may object since the orange color and the cyan color seem to share this same green component. So in fact, whether the green color contributes positively or negatively to the cell's selectivity to color depends on the relative magnitude of this term versus that term. In a special case, if they exactly cancel each other, then this neuron actually has an opponency between just red and blue. However, let's just keep our creativeness in our color naming to get a general gist. Now, if we look at the second output node, O2, then we should pay attention to the second sign here, negative for the red channel and positive for the green channel. And now we see that this positive term, this positive term, these three terms share the same positive sign. And so it's mostly green and with a little bit red. So you can say this is green, yellowish, or lime color. Yeah. And here, this term and that term, they both negative share the other sign negative. And so red and blue together, it's magenta. Yeah. And so this neuron, O2, should exhibit an opponency between lime and magenta. As you can imagine, there can be many other possibilities depending on these gain factors and on these phase values. Therefore, there should be a diverse type of color selectivities in V1 neurons with preferences to various different colors. And this is indeed the case in experimental data. We can also look at the third output neuron, O3. After we work it out, this will be the outcome. Okay, it has the same selectivity to the red and green cone inputs. Okay, so this will be the amplitude and phi will be the phase for red and green inputs. So this will be for the yellow inputs because red and green have the same selectivity. And for the blue inputs, here is the amplitude and that's the phase. So you can see for the yellow input, it's a mixture between luminance filter and a blue-yellow filter. And for the blue input, it's a mixture of luminance filter, another mixture, okay, opposite sign, and a blue-yellow filter. So you can see that it's a multiplexing the luminance and blue-yellow filter for the third output node. And therefore, what we have learned previously when we look at a dichromatic case involving red and green color, and how they multiplex and get our neural response properties, we can borrow the same knowledge and just use it here. Replace red and green color by blue and yellow color. We can also notice that as the gains for the luminance channel becomes stronger, so as this luminance filter becomes stronger or weaker, this can occur, of course, as we go to a higher spatial frequency band. It becomes stronger, and lower spatial frequency, it becomes weaker. And then the neuron's preferred color will mix in more or less of the luminance component. Yeah. So, for example, if a neuron is preferring a red color, when you're mixing more or less luminance component, the preferred color can become pinkish or brownish accordingly. If V1 also have this multiplexing transform that is not the inverse of this original decorrelation matrix, then you can imagine there could be many other possibilities.